So I'm a little bit too fidgety to like stand behind the podium, so I'm gonna like walk around, so I apologize for that. I see my slides over here, but not up here. So unfortunately, I have no punchline for this. So if you're expecting a joke, you might as well just kind of leave if that was what you were thinking. But just to explain sort of the title, but before I do, I just wanted to thank Josh and Joy and everyone who's involved in organizing this awesome symposium because it's been such a great day. And so just to explain, for those of you who don't know me, I'm Vivian. I'm an ocular pathologist, and as an ocular pathologist, two of the most common specimens that I actually review are corneas and skin tumors or skin biopsies. And what I've noticed is that corneas often are taken out not because of tumors, but often because they have visually significant scarring. However, on the other hand, if I look at the cutaneous biopsies, most of them are actually excised due to tumors and rarely due to any wounding. So you can see here an example of a cornea that's part of the enucleated cornea, and you can see that there's a very poor healing scar right here. But if you see these images, which I have to shout out to my very talented, surgically talented colleague, Dr. Brasenyo, you can see that this person underwent a very large excision of a cutaneous tumor, but just even a month afterwards, it healed with very little loss of the integrity. So there's a lot of overlapping processes between tumors and wound healing. For example, both of them are about proliferation, migration, but one is skewed more towards differentiation, while the other is more skewed for undifferentiation. And I'm not the first one to actually figure this out. Dvorak in the 80s actually coined the statement, tumors wound that do not heal. So given the overlapping commonalities, but key differences, I asked myself, is there some sort of mechanism that we can actually harness from wound healing and tumorigenesis that can provide insight for one versus the other? So what do we know? So as just the three objectives that I hope to convey to you is one, to introduce to you SFK as a potential character in both wound healing and tumorigenesis. Two, to cast corneal nerves as influencers in epithelial wound healing and oncogenesis. And then three, explore sort of the role of SFK as a potential hero to rescue wound healing in the setting of neurodegeneration. So if we look at what we already know about epithelial wound healing, when the epithelium is wounded, there's actually release of ATP, which then activates purinergic receptors such as P2YR that releases intracellular calcium in the cell. That actually activates the atom protein to release its pro-ligand, like HBEGF, that then actually starts to activate either in itself or in a neighboring cell, the EGF receptors. Now, there's been a lot of studies that focus initially on EGF receptors. However, it was quickly found out that there wasn't a lot of room to modulate EGFR activity, specifically because they're usually occupied almost the entire time, even at baseline. So given the fact that there's not a lot of room for you to modulate the level of EGFR activity, it was very hard to actually modulate and affect wound healing. Instead, if we look a little bit more downstream, one of the most common downstream from EGFR is SFKs. So SFKs, which stands for Stark family kinases, is a group of 9 proteins. FIN and SARC are 2 of them, which I'll bring up later on in the talk. But they're ubiquitously expressed tyrosine kinases that are non-receptor bound, and it's known to be an effective downstream effector from EGFR. However, unlike EGFR, SFK activity can actually be modulated either by recycling of the SFK or by affecting sort of their negative regulators like sarcasm. I did not come up with that name, that was my mentor's. So it stands for Stark activating signaling molecule, but we can actually modulate the levels. So we asked ourselves, can we actually affect SFK activity to then modulate or impact wound healing? So one of the first experiments that we did was a scratch assay using primary human corneal epithelial cells that we wounded with a shark's tooth comb, and then we pharmacologically inhibited using dasatinib. Dasatinib is a tyrosine kinase inhibitor where at very low concentrations, such as 0.1 nanomolar and 1 nanomolar, as you see up here, is pretty specific for inhibiting SFK activity. And what you see here is that we took images of the cells every hour and measure the area that's filled. And you can see here in the top blue line is the control, and you can see that wound healing is delayed in a dose-dependent manner. And at 24 hours, we see that it's statistically significant, that difference. However, if we look at where the curves really diverge as early on, we can see that even at 6 hours, they're statistically significant. And so this is a time point that I like to draw your attention to. But before I do that, we wanted to tease apart a little bit of what are the cellular processes that are going on that's contributing to that delayed wound healing. So we performed the proliferation and migration assays, and what we can see here is that, again, the dasatinib-treated cells actually showed lower proliferation as well as lower migration rates versus the untreated cells. And so this is probably one of the mechanisms that contribute to that delayed wound healing. We also did a cell death assay, which I'm not showing here in the interest of time, which was actually no difference across the different cell types. So now if we take a look back at the mechanism, right, we can say that we focused on PDK1, S6 kinase, and ERK1/2 because SFKs really affect the downstream effectors of EGFR, and we know that these are downstream effectors. And so when we took a look at this, we did the scratch assay again, but then collected the protein and subjected it to Western blot. And what we see here is what we expect, which is an induction in the level of SFK early on, and then around the same time, also induction of phosphorylated ERK. And then later on, we can see that the PDK1 and phosphorylated PDK1 also increases at 8 hours, and P60, S6 kinase is also increased a little bit later on. So at least we know that these sort of downstream effectors are probably involved, at least when we lower SFK levels. However, we also looked at PI3 kinase as well as AKT, and we saw that there was actually no difference. So, and despite like checking both phosphorylation sites for AKT, it was still non-significant between the different cell types or the treated cells. And so what our data suggests is that at least in terms of pharmacologic inhibition of SFK, that at least we're affecting PDK1, S6 kinase, and ERK1 and 2. However, tumors also exhibit increased SFK activity. And it makes sense, right? Because SFKs are really part of key regulators of cell growth and differentiation. And increasing SFK activity drives carcinogenesis, and they've been found across a whole bunch of different solid tumors. So this seems to be a common driver. But potential mechanisms could include both activating mutations and or sort of impaired down-regulatory mechanisms. However, when they took a look across all of these solid organ tumors, they rarely detected mutated SFKs. So that led us to check whether or not there's downregulation of the negative regulators. So C-Cybl and sarcasm are both negative regulators of SFK. You met sarcasm, but C-Cybl is another downregulator for SFK activity. It's a ubiquitin-mediated proteasomal degradation, while sarcasm actually is an SFK substrate that degrades it through a lysosomal dependent manner. So given that there's increased SFK activity in a lot of tumors, we asked ourselves, well, is there increased SFK levels also in ocular surface tumors, right? So this is an example of a conjunctival intraepithelial neoplasia, and going from left to right, we have unremarkable epithelium going to tumor, and what we can see is that there's stronger staining of SFK or activated SFK over on the tumor side, and less so on the unremarkable side, which is inversely proportional or inversely related to sarcasm, which is much more higher intensity, staining intensity on the unremarkable side and lower in the tumor. And when we compare this also to proliferation rates, we can see that there's an inverse relationship between Ki67, which is a marker of proliferation, and sarcasm. So we asked ourselves, well, if this is a potential mechanism that can be involved with epithelial tumors, which one might be better in terms of a therapeutic target? So we took COS cells, which are immortalized epithelial cells, transfected them with plasmids that either co-expressed activated FIN, which we represent here as FINY528, and either C-Cybl or sarcasm, and then also native FIN and SARC. And I know that this is a confusing sort of slide, But I want to draw your attention to these sort of last columns. So we took these cells and gathered the protein and did a Western blot. And what we can see here is that when the cells co-expressed the mutated fin and C-Sybil, that it was very efficient at downregulating the SFK. However, if we look at sarcasm when it's co-transfected with the FIN-Y528, we actually see that it's not as efficient. However, the story reverses when we look at sarcasm and native SFKs. So here again, I just want to draw your attention to the last two columns, and you can see here that when we have the cells co-transfected with native fin or native sarc and sarcasm, you can see that it's much more or less than when it's C-Sybil. So sarcasm preferentially downregulates native SFK. And so this suggests that maybe tumors, the sort of oncogenetic mechanism that they might use is actually downregulating sarcasm. And so there's a preference of downregulating native SFKs to mutant SFKs. Now, this is the nerve segment. And so you might be wondering, how does this connect to the nerves? Well, clinically, it's already been known, very well established, that corneal nerves are really important for epithelial homeostasis and wound healing. So here on the left-hand side is a clinical picture of those who know of verticillata, which is sort of this whorl-like pattern that you can see in patients taking maybe specific medication. And this sort of swirl-like pattern really follows sort of the migration of the epithelial stem cells to the central cornea. And what we see is that that sort of migratory pattern really reflects or is symmetrical with a corneal nerve sort of swirl-like pattern. So there must be some sort of crosstalk between the nerves and the epithelium at homeostasis. But also when we have mechanisms or conditions where you have loss of the corneal nerves, such as from diabetes or herpes, that there's actually poor wound healing. And so this is an example of neurotrophic keratopathy where this person has a persistent epithelial defect, more because of the sort of epitheliopathy that has formed from the dysfunctional nerve. There's also increasing evidence that peripheral nerves contribute to tumor invasion and metastases. This is actually an example of an adenoid cystic carcinoma that I received, and you can see here that there's perineural invasion right here, and it's thought that the nerves actually produce like sort of a micro niche that helps the tumor cells migrate and invade further in the tissue. So we wanted to study this a little bit further, and so we we inherited a model of neurotrophic keratopathy where we isolated the ciliary nerves as it straddles the optic nerve and then pierces the posterior sclera, and we transected them, and what we did was we performed the transection on one side and then a sham surgery on the other side, then created an epithelial wound, and then measured the remaining defects 16 hours later. And as you can see here, these are representative photographs. And this is just preliminary data, but you can see at least that there's a trend that follows what we see clinically, which is there's much more faster wound healing in the sham eye versus the transected eye. However, what I just told you about SFK is that modulation of SFK can actually potentially modulate wound healing, right? And I showed you the opposite where it's like decreasing SFK levels can delay wound healing. Well, can the opposite actually help recover the wound healing and denervated nerves if we increase SFK levels? So what we did was we compared wild-type mice to a mouse model that overexpresses FIN, which is FIN-Y528. As you can see here in this represented sort of picture, we have the transected nerve on one side and then the sham eye, and we compared wild-type to FIN-Y528, which is that mouse that overexpresses SFK, and measured the remaining epithelial defect at 6 hours. And what we see graphically, this is, and I'll walk you through this, is that one, as we expected, there should be faster wound healing. Oops, that's why. Faster wound healing in the wild type, right, compared from the sham eye to the transected eye. But more importantly is that in the FIN-Y528 mouse, you actually see that there's no statistically significant difference between the sham eye and the transected eye. But even more surprising is the fact that there's no statistical difference between the eye, the wild-type eye that underwent the sham surgery and the FIN-Y528 that underwent the transected nerves. So seeing this sort of observation that there's some recovery of the wound healing from increased SFK levels means that that overexpression or sort of like making the epithelium a little bit more robust can actually help offset some of these effects from a denervated cornea. Interestingly, I'm not going to say the name because I always kind of mess it up, but there's a recombinant form of nerve growth factor that's the only FDA-approved medication to treat neurotrophic keratopathy, and that also targets the corneal epithelium. So hopefully, at the end of this presentation, I've successfully introduced SFK as a potential character in both wound healing and tumorigenesis, also introduced corneal nerves as an influencer in epithelial wound healing and oncogenesis, and then hopefully sort of give you a taste of SFK's ability to rescue wound healing in the setting of nerve degeneration. And I'd like to acknowledge my lab members as well as my collaborators and members of their lab, as well as my funding resources. And the only thing that I would like to point out is that we also have a collaboration with 2 other ocular pathologists where we have an Instagram account where we post really interesting ocular pathology cases. So please visit us. That's the handle. Thank you so much for your attention. Happy to take any questions.
Any questions? So
I was wondering about the swirling pattern in the eye. I thought these were mitotic cells maybe typically, but you're saying they can be induced by medication?
So the medication is actually, it's not, so certain like medications like amlodipine, they produce a metabolite, then you can actually see. So these are transient amplifying cells from the stem cells that just get the metabolite, and so you can see that verticillata, yeah.
So can you just reiterate for us, sarcasm is good for wound healing and bad for tumors? Or do I have that backwards?
No, so sarcasm is good for tumors, bad for wound healing.
And
don't ask me for the punchline for the joke.
no, how about the punchline for the clinic? How available are these kinds of inhibitors versus activators? How do you envision promoting corneal wound healing with this knowledge?
So actually, part of the thing, I was trying to get these sort of images. So we're actually investigating the role of AAV vectors, and so because they're episomal, they're not lasting. And so if we can inhibit sarcasm or promote SFK activity through AAVs, it can be a potential therapeutic modality that we can use. But I mean, it's still gene therapy. It's not like an antibody or a small molecule.